Okay, great. I'd like to get started. Thank you all so much for joining us today at the Connect to Science seminar series. A couple of quick reminders before we get started today. I'd like to encourage you all to visit the website listed at the bottom of the title slide. That's a site where you can learn about upcoming speakers and get other updates on the Connect to Science series. Also, during the course of the presentation, please post questions in the Q&A section at the bottom of the screen instead of using the chat box. That'll just make it easier for us to coordinate the question and answer session. So today I'm so pleased to introduce Professor David Kirsch from the Duke University Medical Center as today's speaker to the DFCI Connect to Science series. David's been a leader and true visionary in the fields of radiation oncology and radiation biology, and has greatly enhanced our understanding of how tumors respond to cancer therapies with a particular focus on ionizing radiation, as he'll likely tell you today. This is a bit of a virtual homecoming for David, who received his MD and PhD degrees from Johns Hopkins, but then came to Boston for his residency training in radiation oncology at Mass General and Harvard Medical School, and then stayed on to do a postdoctoral fellowship in the lab of Tyler Jacks at MIT. During that time, he used genetically engineered mouse models to make some really cool and important discoveries related to the effects of ionizing radiation on healthy as well as cancerous cells. Professor Kirsch is a bona fide physician scientist in every sense of the word, with his time evenly split, essentially, between making impactful discoveries in the laboratory and also leading the clinical trials that bring innovative treatments directly to patients. David is also a tireless advocate of his trainees, who I've met many of, and has received the Dean's Award for Excellence in Mentoring from Duke University. That's probably one of the awards that he's most proud of in his illustrious career. So without further ado, I'd like to turn the floor over to David. He's going to be telling us today about some of his insights into how tumor cells respond to cancer therapies and immunotherapies in particular. We're so glad you joined us today, David. Looking forward to your talk.
Chris, thanks so much for the kind introduction. Can you see my slides and also hear me okay?
Yes.
Great. So real pleasure to get to be here virtually with you and tell you about some of our recent work studying mechanisms of sarcoma response and resistance to radiation and immunotherapy. As Chris mentioned, I'm a radiation oncologist, physician scientist, and my clinical practice focuses on sarcomas, patients with bone and muscle and other connective tissues. And also my laboratory research uses genetically engineered mouse models as well as human tissues and cell lines to study sarcoma biology and radiation biology. So this is my disclosure slide. None of this will be relevant to the talk today, except that I will present an update of a clinical trial that is supported by Merck. So sarcomas are a complex heterogeneous group of tumors. They make up over 70 different subtypes. And broadly speaking, if you look at them genomically, they can be categorized into two groups. One group, probably about 2/3 of the various different types of sarcomas, have copy number alterations, kind of a very complex aneuploid genome characterized by p53 mutations. And the other 1/3 of soft tissue sarcomas are characterized by fusion oncoproteins. And this is generally, as shown on the surface plot, more of a simple karyotype that's present. And so for today's presentation, I'm going to talk about one of the fusion type proteins drivers of sarcoma, myxoid liposarcoma, driven by the Fus-CHOP translocation. And this really piqued our interest because this type of sarcoma has exquisite radiosensitivity. Then I'll give you an update on our clinical trial, Stand Up to Cancer SARC-32. It's a randomized clinical trial testing the combination of preoperative radiation therapy and the anti-PD-1 agent pembrolizumab. And then I'll spend the rest of the presentation talking about our novel high mutational load sarcoma mouse model and the differences between the response of transplanted tumors and the primary sarcomas from this model to radiation therapy and immunotherapy. So let's start with myxoid liposarcoma. This was first described as a very radiosensitive tumor by James Ewing, the esteemed pathologist working in New York over 85 years ago. And here in this case report, he described a middle-aged woman who had a bulky tumor of the false pelvis. She was treated with 125 kilovolt X-rays, so not even the megavoltage X-rays that we use today. And the structure of this tumor was a myxosarcoma, kind of the predecessor to what we call myxoid liposarcoma. And he noted that the tumor in the pelvis disappeared rapidly under irradiation. And this is really unusual for sarcomas. Usually the tumors kind of sit there, some of them swell with inflammation, but it's very unusual for a sarcoma to kind of melt away with radiation therapy. So here's a more contemporary study done by Peter Chung and colleagues from the Princess Margaret Hospital in Canada. This is the classic histology taken from a biopsy of a patient with a myxoid liposarcoma in the leg. You can see this very bright myxoid tissue that's showing up here on the T2 MRI. And you can see this chicken wire pattern of the vasculature that's present in this background of myxoid and tumor cells. The patient had 50 gray of radiation therapy given at 2 gray daily fractions. You can see on the MRI marked regression of the tumor. And then when the tumor was resected, you can see almost all of the tumor cells are gone. The vasculature's been depleted, and you're left with this kind of this empty tissue left behind. So marked regression to radiation therapy. And Pitson and colleagues actually quantified the changes in volume by MRI of these tumors. Here's for comparison, undifferentiated pleomorphic sarcoma, one of the most commonly diagnosed sarcomas in adults. And you can see that the volume really doesn't change very much. Some of the tumors are actually getting bigger on radiation therapy. A couple are getting a little smaller, whereas for the myxoid liposarcomas, they see that there's regression occurring for these kinds of tumors. Now, this marked radiosensitivity translates into excellent local control. Here's the team from Toronto showing that local control rates at 5 years after 50 gray of preoperative radiation therapy followed by surgery are in the 97 to 99% range. Ashley Guadagnolo, who's a graduate of the Brigham and Women's Dana Farber residency in radiation oncology, now leading the sarcoma program at MD Anderson in radiation oncology. She also reported outstanding local control for myxoid liposarcoma following the standard 50 gray of radiation therapy following surgery. And because the outcomes are so good, there's now an effort to try to de-intensify therapy. Rick Haas and colleagues from the Netherlands Cancer Institute published the DOREMI study where they did dose-reduced preoperative radiation therapy in myxoid liposarcoma. This was a single-arm study of 79 patients with myxoid liposarcoma, and instead of the standard 50 gray, these patients all received 36 gray and 18 fractions. And the median follow-up still a little bit on the short side at 25 months, but you can see they have 100% local control at this time. So really excellent outcomes. And as this study matures further, I think it's likely that 36 gray will become the new standard of care for preoperative radiation therapy. So, why is this tumor so sensitive to radiation therapy? Well, the driver gene for myxoid liposarcoma is the FUS-CHOP fusion protein. And here's the schematic of the various components of the FUS-CHOP fusion protein at the top. This comes from the DDIT3 gene, which encodes the FUS or fused in sarcoma protein. This has an N-terminal prion-like domain followed by a G-rich area. There's a translocation that happens within the DDIT3 gene, and it is fused to the transcription factor CHOP. So normally CHOP is a gene that gets induced with ER stress and binds to CHOP binding sites to drive various transcription of important genes to respond to ER stress. But this has a DNA binding domain and a leucine zipper shown here. But the translocation happens between the G-rich protein of a G-rich area of FUS and the 5' untranslated domain. And then there can be a fusion protein that can be made that drives myxoid liposarcoma. So FUS is part of a family of genes that are translocated in sarcoma, part of the FET family, which in addition to FUS, there's EWS, which occurs frequently in Ewing sarcoma, as well as TAF15. And the various types of fusion driver sarcomas that can arise include myxoid liposarcoma, Ewing sarcoma, desmoplastic small round cell tumors, angiomatoid, fibrous histiocytoma, and extraskeletal myxoid chondrosarcoma. And as you can see here, for a myxoid liposarcoma, the pathognomonic fusion is FUS-CHOP, but in rare cases, actually, EWS can fuse with CHOP. So EWS can replace FUS and drive myxoid liposarcoma. Similarly, whereas for Ewing sarcoma, EWS is the pathognomonic fusion partner for an ETS family transcription factor, in rare cases, EWS can be replaced by FUS as the N-terminal driver. And the same can happen for angiomatoid fibrous histiocytoma, that there can be a swapping of EWS for FUS. And so these data really imply that FUS may be driving cancer in a similar way to EWS because they can substitute for each other. And there's been a lot of work, a lot of beautiful work coming out of Dana Farber and other institutions studying EWS-mediated transformation, as well as other as well as other fusion proteins that drive sarcoma. And I'll just highlight some of this work briefly. So for Ewing sarcoma, for example, work from Sigal Kadosh and Miguel Rivera, both from Harvard, published this really important paper on Ewing sarcoma showing that it can retarget BAF complexes mediated by this N-terminal prion-like domain. And then Scott Lowe and colleagues at Sloan Kettering, looking at synovial sarcoma and the SS18 SSX oncoprotein, showed that it could hijack the polycomb repressive complex and again to modify chromatin as a means to drive synovial sarcoma. And then Beat Schaefer from Zurich and his colleagues there studying alveolar rhabdomyosarcoma and the PAX3 FOXO translocation showed that the CHD4 helicase, which is an epigenetic co-regulator, could also be a key mechanism for sarcoma development. And so building off of this kind of data, we thought that maybe FUS was also interacting with chromatin modulating proteins. I just want to show you one slide taken from the Boulay paper, and because this really informed our thinking about how to study FUS-CHOP and myxoid liposarcoma. Here we're looking at chromatin immunoprecipitation sequencing data. So we've got a chip to FLY1 pulling down EWS fly, showing that it's binding to these GGAA repeats. And then there's this is the SHGFP control cells. When the Ewing sarcoma cell lines are infected with a virus expressing SH to EWS fly, and to knock it down, you can see that you lose those binding sites there on the GGA repeats. And then BAF157, one of the key proteins in this BAF complex, is also being recruited to the same sites shown here in the SHGFP Ewing sarcoma cells. But then when there's an SH-EWS FLY1, so there's a knockdown of EWS FLY1, then you lose the targeting of the BAF complex. And this is just shown here schematically, where you have closed chromatin normaling along these GGA repeats. Transcription of important genes that can promote sarcomagenesis are off. But then in the presence of EWS FLY1, there's binding to this active enhancer, changes to the chromatin complex as a result of the BAF complex being recruited, and then this can turn on key target genes. So our hypothesis kind of going into this project was that FUS-CHOP and myxoid liposarcoma could also interact with various chromatin remodeling complexes via the prion-like domain to activate an oncogenic transcriptional program. And then we also thought that perhaps radiation therapy could somehow disrupt this. And one hypothesis that we had was that with ionizing radiation, there's activation of DNA damage response kinases, and these could phosphorylate the N-terminus of FUS, and this could then alter the protein-protein interactions of the chromatin interacting complexes. And here just are some of the ones that we studied. The BAF complex shown here. There's also the imitation switch complex with SNF2H binding to ACF1 here. And then there's also the NERD complex with CHD4 that can bind to HDAC1. And so this is the work of Mark Chen, a really talented MD PhD student who's now here at Duke for his radiation oncology residency. And so Mark did some proximity ligation assays with antibodies to SNF2H, FUS-CHOP, and you can see here in the 2 human myxoid liposarcoma cell lines, you can see these bright red dots showing that there's close proximity between these 2 proteins. As a control, there's the SW872 cell line. This is a pleomorphic liposarcoma human cell line that does not express the FUS-CHOP protein. So of course, it's a nice negative control. We also saw co-localization of the antibodies with BRG1 and FUS-CHOP, and then to a lesser extent, CHD4 and FUS-CHOP, and the data is quantified on the right. Now, then Mark wanted to try to validate this interaction doing co-immunoprecipitation studies, again, taken from the same human sarcoma cell lines. So here are the antibodies to SNF2H or an IgG control. You can see SNF2H in all of the cell lines is coming down with its partner protein ACF1. But only in the myxoid liposarcoma cells are we seeing the FUS-CHOP band come down. And this is, you can see that the proteins can migrate at 2 different places here because the fusions for these 2 patients have different variants. So the translocation's happening in 2 different parts of the genome. And so there's a 7.2 variant or the 13.2 variant. Both of them can interact with SNF2H. Similarly, both of these variants of FUS-CHOP can also interact with BRG1 in the 2 human myxoid liposarcoma cell lines that we were able to get. However, Mark was not able to confirm an interaction between CHD4 and FUS-CHOP. So we think that this may have just been a spurious result from the PLA experiment, but these other 2 interactions with these chromatin remodeling complexes are confirmed by co-immunoprecipitation. So then Mark went on to do ChIP-seq experiments, looking at FUS-CHOP, SNF2H, and active H3K27 acetylation. And you can see now we were hoping to see binding of FUS-CHOP to GGA repeats or something novel like that, but instead we saw that FUS-CHOP bound to CHOP binding sites. And this was specific, of course, to the 2 cell lines that express FUS-CHOP. Similarly, we could see targeting of SNF2H to the same sites in both cell lines. And we also saw active H3K27 acetylation at these sites as well. This is just kind of zooming in to one of the binding sites for FUS-CHOP shown here. This is the control cell line, control liposarcoma that doesn't have FUS-CHOP, and you can see there's no SNF2H or H3K27 acetylation at that site. In addition, with RNA sequencing, we could see that the enhancers would upregulate a common set of genes, specifically in the 2 myxoid liposarcoma cell lines. And I'll just kind of summarize here before moving on. The data that I've shown you so far indicates that FUS-CHOP can interact with multiple chromatin remodeling complexes. And for example, SNF2H can be retargeted to FUS-CHOP bound enhancers in myxoid liposarcoma cells to drive transcription. And so then we wondered about, well, how would this model then inform the exquisite radiosensitivity we see with myxoid liposarcoma? So it turns out that it's known that FUS can be phosphorylated on its N-terminus within the prion-like domain. There are SQTQ sites, so serine glutamine or threonine glutamine sites that can be phosphorylated by various DNA damage response kinases that include ataxia-telangiectasia mutated and DNA PK, so some of the key kinases that are induced by double-strand DNA breaks. And so one possibility that we considered was whether phosphorylation at this N-terminus could impact the protein-protein interactions that we defined. And so we first set out to see, could these SQTQ sites be phosphorylated? So Mark did experiments where the cell lines would be treated with and without ionizing radiation. For this first experiment, we're looking at 5-gray. Then he did IP immunoprecipitation for CHOP, pulling down FUS-CHOP, and then could use an antibody specific for phospho-SQTQ, and could see that in the presence of ionizing radiation, there would be an induction of the ability of this phospho-SQTQ antibody to bind. He could then use inhibitors to either DNA PK or ATM, and this blunted the ability of radiation to phosphorylate these sites. And here's a specific antibody now for the S42, serine 42 of FUS, giving this, this is a mouse cell line that expresses FUS-CHOP. With 10-gray of radiation, we can see the controls, CAP1 being phosphorylated here, an ATM target. And we also can see that serine 42 of FUS-CHOP is now being phosphorylated. And then in the presence of an ATM inhibitor, we see diminished phospho-CAP1 on the Western blot. And similarly, the phospho S42 band is now gone away. And we also have a KP mouse sarcoma cell line that doesn't have FUS-CHOP as a control. Similarly, Mark did co-immunoprecipitation and could see that irradiation would diminish the interaction of FUS-CHOP, SNF2H by co-immunoprecipitation. Here showed with 10-gray of ionizing radiation, there's less FUS-CHOP that comes down in the co-IP, and this is now quantified here on the right. So then Mark made some site-directed mutagenesis of all of the potential sites of phosphorylation in the prion-like domain. And so here we have the wild-type FUS-CHOP construct shown here. He made a 12A mutant where alanine replaced all the serine and threonines. So this is a phospho-dead mutant. And then he also made a 12E mutant where all of the 12 serine threonines are mutated to glutamic acid as a phosphomimic mutant. He expressed either the wild-type FUS-CHOP or the phospho-dead and phosphomimic mutants in NIH3T3 cells. There was no effect on the growth of these cells in vitro. However, there was a marked effect on the ability of FUS-CHOP to interact with SNF2H. So here you can see that the wild-type protein interacts nicely with SNF2H. The 12A mutant, the phospho-dead mutant, interacts well with SNF2H. But when there's a phosphomimic, as if all of the phosphorylation sites have a phosphate group on it, now you can see that there's disruption and there's no longer good interaction of SNF2H with FUS-CHOP by the co-immunoprecipitation assay. Mark then also looked to see what would happen when FUS-CHOP was expressed in the NIH3T3 cells. He found that this was sufficient to transform them as an assay by soft agar growth. Here we can see FUS-CHOP being sufficient to form colonies in soft agar. The 12A mutant, the phospho-dead mutant, was able to also form colonies. However, the phosphomimic 12E mutant failed to form colonies in soft agar. And then Mark also wanted to do a rescue experiment. So here he did an shRNA to FUS-CHOP. shRNA1 worked well, and number 3 worked pretty well in addition. And so he was able to take the myxoliposarcoma human cell lines, knock down FUS-CHOP. This impaired the ability to form colonies in soft agar. However, he could then rescue this defect with the 12A phospho-dead FUS-CHOP that could not be knocked down, shown here, so we could reform colonies. However, when he did the exact same experiment with the 12E mutant, so this is the phosphomimic mutant, he was not able to rescue the knockdown phenotype. And so it's important that to have an unphosphorylated state for FUS-CHOP to be able to exert its ability to transform the cells. What's the mechanism of this phosphoregulation and interacting with the proteins? Well, we had a hint that this might be potentially due to phase separation. And so to start to look at this, Mark generated an additional fusion. So here's the FUS-CHOP with a maltose binding protein to provide solubility and a tobacco etch virus protease cleavage site as a linker. So you can see that wild-type FUS-CHOP is very soluble, When you add the TEV protease to cleave off the maltose binding protein, then you form particles in approximately the size range. The 12A mutant, also with cleavage of the protease, again forms nice particle sizes here. But the 12E phosphomimic, you can see even after cleavage remains very soluble. And this data is really consistent with work from Izzy Owen at the NIH, where she did experiments, again, looking for FUS-CHOP. This is the type 2 version of FUS-CHOP, where she could see that with cleavage of the TEV protease, she could see that there were droplets that were formed. And she also saw that these kinds of phase separation could occur in the nucleus of myxoliposarcoma cells in the actual cells with live imaging. So taken together, all of this data suggests a model for sarcomagenesis and radiation sensitivity, where we have FUS-CHOP binding to proteins like SNF2H to help drive sarcomagenesis. Then there can be ionizing radiation causing phosphorylation of the N-terminus prion-like domain that interferes with some of the chromatin remodeling complex interactions. In addition to radiation causing double strand breaks and killing the cells through a standard mechanism, the radiation here is interfering with a key mechanism that helps to maintain the tumor in a cancer state. So again, just to summarize this part, we find that FUS-CHOP can interact with chromatin remodeling complexes and retargets SNF2H and others, including the BAF complex to FUS-CHOP bound enhancers. Phosphorylation of the FUS-CHOP prion-like domain can decrease interactions with SNF2H and also inhibit transformation by the fusion protein. We also find that there's increased solubility of the phosphorylated form of FUS-CHOP, and we think that that may underlie the exquisite radiosensitivity that we see in patients with myxoid liposarcomas, perhaps by disrupting these critical protein-protein interactions that are needed for sarcomagenesis. So now I'm going to turn to a different type of sarcoma. We're going to talk about undifferentiated pleomorphic sarcoma now, and just give you a brief update of our Stand Up to Cancer SARC-32 clinical trial studying radiation therapy and immunotherapy. And our clinical trial really started based on this study by the SARC-28 team. So SARC stands for Sarcoma Alliance for Research Through Collaboration, which is a cooperative group that studies sarcomas in clinical trials and also helps to support translational research. The SARC-28 investigators studied 40 patients with soft tissue sarcomas with metastatic sarcoma and looked at the anti-PD-1 agent pembrolizumab. And the SARC-28 investigators were very thoughtful. They didn't just enroll all 70 plus types of sarcomas. They wanted to look at 4 of the most common soft tissue sarcoma subtypes for this study. So they enrolled 10 patients with leiomyosarcoma, sarcoma of the smooth muscle lineage, undifferentiated pleomorphic sarcoma, which is most likely from the skeletal muscle lineage, liposarcoma from the fat lineage. So these 3 are all of the kind of aneuploid copy number variant sarcomas. Then there's synovial sarcoma, one of the translocation-driven soft tissue sarcomas. And what they reported was that they had a 40% response rate for undifferentiated pleomorphic sarcoma. And then for liposarcoma, these aren't the myxoid liposarcomas here, but this is the dedifferentiated liposarcoma or pleomorphic liposarcoma. They reported a response rate of 20%. And so as we move forward, thinking about designing of our study, we wanted to kind of focus on the sarcoma subtypes that showed a signal to pembrolizumab in the metastatic setting. And so here, this is one patient who had a metastatic undifferentiated pleomorphic sarcoma. You can see really responding well to pembrolizumab. Here, the red color shown here on this waterfall plot, the undifferentiated pleomorphic sarcomas are in this kind of maroon color here, responding well. And the green are the poorly differentiated or dedifferentiated liposarcoma patients. And Petitprez and colleagues collaborated with the SARC-28 investigators to identify a sarcoma immune class that was associated with immunotherapy response. And this is looking at gene expression profiling data here. And you can see that the tumor types here that have a red color are enriched for an immune signature with T cells, B cells, and other immune cells. And they call this immune class sarcoma immune class E in comparison to the A, B, C, and D, the other 4 subtypes. And now when in their paper published in Nature, where they have this waterfall plot, now the colors are based off of the sarcoma immune class. And you can see that the tumors that are responding the best, these 3 undifferentiated pleomorphic sarcomas in red, these are all the sarcoma immune class E tumors. So there may be certain subtypes of sarcomas that have a tumor immune microenvironment that makes them more likely to respond to immunotherapy. Now, our work with SRC32 was really to test the idea that radiation with immune checkpoint inhibition may have synergy. And this work really was built on a number of publications from many different groups around the world. Here, I'll just show one. This is the work from Andy Min's group at Penn, where they looked at the B16 F10 melanoma transplant tumor model, and they saw that when they gave radiation therapy with anti-CTLA-4, they saw a marked improvement in response that they didn't see with any of the treatments alone. Here's an example here in another experiment where they used radiation with anti-PD-L1 with anti-CTLA-4, seeing much better responses than single treatment alone. And so taking all of this data together, we move forward with Stand Up to Cancer SRC32, where we're enrolling patients with intermediate or high-grade undifferentiated pleomorphic sarcoma or pleomorphic dedifferentiated liposarcoma. The tumors have to be large, greater than 5 centimeters. Eligible patients are stratified by grade and randomized to a standard treatment, which is our image-guided radiation therapy, 50 gray and 25 fractions, followed by surgery, and then the patients are followed. And then the experimental group get pembrolizumab before, during, and after radiation therapy. And then after surgery, there's another adjuvant pembrolizumab up to 14 cycles, so 200 milligrams every 3 weeks. And this study is really rich and correlative. So we have upfront pretreatment biopsy samples, we have resection samples on all the patients, and we also have blood samples before, during, and after radiation therapy, and also 2 blood samples after surgery as well. I want to just mention this as a huge effort across the US and in other countries. We have 13 US sites enrolling patients, sites in Canada and Montreal, Milan, and 3 sites in Australia are all accruing patients. We now have 103 patients enrolled with a target accrual of 126. And I want to give a big shout out to the Dana Farber Cancer Institute, Mass General sarcoma teams, because you guys are the #1 accruer so far in the US, but I want to note that University of Michigan's catching up. So we still have 23 slots to go. So definitely want you to keep on accruing these patients, because we're getting close to the end of this important study. So I just wanted to give you a sneak peek at some of the correlative data that we've got so far. This is early, it's ongoing. We don't have data on which patients are assigned to pembrolizumab. The outcomes of the patients are still blinded to us, but we've begun the correlative studies, and I just want to show you what we're doing at this point. Right now, we're comparing the pretreatment biopsies with the surgical resection samples. This is done in collaboration with Everett Moding, who is a former trainee in my lab, now with his own lab and a sarcoma radiation oncologist at Stanford. In the SRC32 samples, Everett's found by RNA sequencing of the upfront pretreatment biopsy. We can see that there's a significant number of these patients that have the sarcoma immune class E gene signature that was identified by Petapres colleagues. And so based off of this, we would think that these patients may be enriched in responders, and we do have a pre-specified subset analysis in our current amended protocol to specifically look by sarcoma immune class to see if this correlates with outcome or not. And what's really nice about our study, because we have a randomization with and without pembrolizumab, we will be able to tell whether sarcoma immune class E is prognostic or predictive of outcome with pembrolizumab from our study. Everett also did cybersort analysis of the RNA sequencing studies. This is a bioinformatics approach to assess the the quantity of the various types of immune cells in the tumors. And here he's comparing the pre-radiation therapy versus the post-radiation therapy resection samples. Some of these patients also got pembrolizumab, but again, we're blinded to that. But we can tell when we compare these two is that after neoadjuvant therapy, we see an increased number of plasma cells, increased number of CD8 T cells, and a decreased number of regulatory T cells and M2 macrophages. Again, we don't know if this is different by randomization arm, whether pembrolizumab makes a difference in this or not. But we're excited that we can see that there are immune changes happening with the neoadjuvant therapy. In addition, there appears to be a distinct microenvironment that may be consistent with a favorable response to radiation therapy with or without pembrolizumab. And so looking at the patients that had a very high rate of necrosis, greater than 95%, compared to what we call low necrosis with less than 95% necrosis in the specimen. We could see those patients that had high necrosis, had high memory B cells, they had regulatory T cells and activated NK cells, but low numbers of activated memory CD4 T cells by the CyberSort analysis. So very early days with this, but we're really excited to finish the trial, unblind the data, and see if pembrolizumab is either helping patients or not, but also to see if there's we're going to learn a lot of important work on human sarcoma biology from the correlative studies. So now for the last part of the presentation, I want to return back to some preclinical work and share with you some of our mouse modeling studies of studying the response of tumors in a transplant tumor model or the primary sarcoma model to radiation therapy and immunotherapy. So, each model system has various strengths and weaknesses. The genetically engineered mouse models that we generate, they're autochthonous or primary tumors that co-evolve with the immune system where there's immunoediting and immunosurveillance that's there. And so that's a real strength of these models. But kind of our workhorse model, the model that I developed when I was in Tyler Jacks's lab, as a postdoc, which has mutations in KRAS and deletions of P53, these tumors have very low tumor mutation burden and by whole exome sequencing. And so there may not be a lot of neoantigens that can engage the immune system. And work from a number of labs over the past 6-7 years has shown that the response of human cancers to immunotherapies improve with high tumor mutation burden. So here's one study by Rizvi and colleagues where non-small cell lung cancers that had high non-synonymous mutations were more likely to respond well to immune checkpoint inhibition. And then mismatch repair deficient colorectal cancers, as you know, respond much better to immune checkpoint inhibition. And even locally advanced cancers now, patients can get complete response in the early studies that are being reported now with immunotherapy. And so a really talented radiation oncology resident, Yvonne Mowry, now with her own lab here at Duke, came to my lab as a resident, and we set out to make a new genetically engineered mouse model of sarcoma with high tumor mutation burden. And for this model, Yvonne took the P53 flox mice where exons 2 through 10 are flanked by LOXP sites. She injected into the gastrocnemius muscle an adenovirus expressing Cre recombinase to delete both copies of P53. And then she also injected in the chemical carcinogen 3-methylcholanthrene into the same muscle. And it takes about 2 to 3 months for the tumors to form. And then Yvonne isolated several of these tumors. And she found that compared to the standard KRAS P53 model, where whole exome sequencing had only about 18 non-synonymous somatic mutations, in the P53 MCA model, the genetically engineered plus chemical carcinogen model, now the median number of non-synonymous somatic mutations was about 2,000. So an order of magnitude, 100-fold more mutation burden. So we assume that this would have plenty of neoantigens to engage the immune system. And then a really talented MD PhD student, Amy Wisdom, came to the lab for her PhD work. Amy's now a Harvard Radiation Oncology Program resident. And Amy took the model that Yvonne generated, and she did what many in the field were doing at the time, which was to take a primary tumor tumor, make a cell line, and then inject it into the muscle of the mouse to make an orthotopic tumor, to make a transplanted tumor. And these tumors are similar to human undifferentiated pleomorphic sarcoma. And what Amy found, just as many others in the field have published, is you can take these cell lines, make a transplanted tumor, and then when you give anti-PD-1, there's a little bit of a response. In this case, she's giving 20 gray of radiation therapy in a single dose, seeing a modest response, but curing a fraction of the animals. Then when she combines radiation therapy together with anti-PD-1, all of the animals are cured. And then Amy did a very important experiment, is that she repeated these studies now in the autochthonous, or the primary tumor model. So this is the exact same model as her cell line model, where she uses the adenocrete to delete both copies of p53, and then also uses the chemical carcinogen MCA, so that when the tumors get a primary sarcoma in the gastrocnemius muscle, now she gives anti-PD-1, there's very little response. Radiation therapy can cause a growth delay, but the combination of radiation therapy with anti-PD-1 doesn't really have synergy now in the primary tumor system. She also did an experiment, similar type of experiment now, using radiation therapy with anti-PD-1 and anti-CTLA-4 antibodies. Again, in the transplant model, she cures all of the mice with tumors. But in the autochthonous model with the p53 MCA primary tumors, the combination of radiation therapy plus anti-PD-1 and anti-CTLA-4, shown here in the red dash, shows that there's very limited efficacy. And so there's a real disconnect between what we see in the response to our transplant models versus the autochthonous models, where the primary tumor co-evolves with the immune system. So for Amy's PhD, she really did a very in-depth characterization and compared the transplant versus the primary models using whole exome sequencing and RNA sequencing, immunohistochemistry, single-cell RNA sequencing, and as well as mass cytometry. And I'll just share a couple of the highlights from this work, because it's all published. Here, looking at the number of non-synonymous mutations, we can see that the tumor for this study that was picked for the cell line work is shown here in black. And then in the transplant model with no treatment, anti-PD-1 treatment, radiation alone, or the combination, there's not a very big difference in the number of mutations that are present. Most of the p53 MCA primary tumors actually started out with a higher tumor mutation burden. So the reason why the transplant models responding and the autologous models not responding isn't due to a difference in tumor mutation burden. However, we did see that under RNA sequencing, our analysis, we did see that there was decreased neoantigen fraction expressed. And so although both of the tumor models have neoantigens based off of the DNA mutations that can be seen by whole exome sequencing, the neoantigen expression is much lower in the primary tumor than the transplant tumor. So there's a less ability to engage the immune system. And then Amy confirmed this by generating primary tumors in RAG2 null immunodeficient mice or RAG2 heterozygous immunocompetent lintermate mice. And for this experiment, she's using CRISPR-Cas9 to delete or to inactivate p53 alleles, and then also giving the chemical carcinogen MCA. And you can see that the number of non-synonymous mutations is actually higher in the absence of an immune system. And then the neoantigen expression level is also much higher in the absence of an immune system. So the data show that there's selective pressure to eliminate some tumor cells. The immune system can eliminate some tumor cells, and also the cells that can grow out are the ones that downregulate the neoantigen expression. And then Amy also did experiments. I showed you that's data showing that there's cell autonomous effects where changes in the tumor cells can affect this tumor immune interaction, but there can also be changes to the immune system. There's immune tolerance. And this was shown by really elegant transplantation studies. So Amy generated cohorts of mice that we call donor mice with primary tumor. She did an amputation of the primary tumor to make a cell line. And then she transplanted that cell line either into naive syngeneic mice or retransplanted it back into the donor mouse. And then when she did this, you can see that the donor mice quickly retook their own tumor back. So 100% of the mice grew a tumor pretty quickly, whereas the naive syngeneic mice in this experiment, only about 50% of them were able to grow out a tumor. And then when she, and with more delayed kinetics, and then when she treated these tumors with that did grow out with anti-PD-1 and 10 gray of radiation, you could see that about 50% of the mice were cured in the syngeneic model in this experiment, whereas none of the donor mice where the tumor grew out that she was able to treat them with radiation and PD-1, were there any tumors that were cured. Now you might say, well, a mouse that has had a tumor and then you amputate it, it's kind of beat up. It's just immune system's going to be unhappy because it's had a cancer. And so as a control experiment, Amy generated another cohort of donor mice where she did an amputation and then had other mice that she got a primary tumor and did an amputation to make cell lines. And so then she could do transplantation back into the donor mouse, either with a self tumor or a cell line from a different mouse, a non-self tumor. And when she did this, the donor mouse that received its own tumor, the self tumor, of course, they all grew in the mice. But when those donor mice that had had a primary tumor that had been amputated, when these mice then received a cell line from a different mouse, syngeneic mouse, they were able to reject all of the tumors. So the immune system in the donor mouse can reject and see foreign tumor cells that are injected in, but they're unable to reject its own tumor. And then finally, she did a bone marrow transplant experiment to see if she could try to reverse this. And so she did total body irradiation after amputating primary tumors from donor mice or naive mice, and then did bone marrow transplantation experiments to see if she could put in fresh bone marrow, if that would affect the ability of the immune system to recognize the transplanted tumor. And you can see here with the tumor onset that now the donor naive mice have a similar tumor take rate. So she's reset the immune system, and then giving anti-PD-1 and radiation, there's also a similar rate of response here in this experiment, suggesting that there's a way, there are ways to kind of reset the immune system to attack tumor cells. And then we also wanted to see whether the transplant tumors and the primary tumors resembled human cancer. And so for this, we collaborated with Max Dean's lab at Stanford. Here are the murine tumors. We're looking at, in red, the transplanted tumors, and in blue are the primary tumors. We're looking here by cybersort analysis, and you can see that the immune cells, the T cells, the B cells, and others are more expressed in the highly inflamed transplant tumors compared to the primary autochthonous murine tumors. And the transplant tumors are most similar to the sarcoma immune class E in human patients. And this is shown here, where looking at the cell fraction of the immune cell content, we see that the transplant tumors are more like sarcoma immune class E compared to the primary tumors, which are more like the other human sarcoma immune classes. And specifically looking at activated CD8 T cells by the RNA-seq bioinformatic analysis, again, we see much more higher CD8 positive T cells in the transplant tumors and the primary tumors. And this is confirmed by immunofluorescence shown here on the right of these transplant versus primary tumors. So I'll just summarize Amy's work here. We see with the primary tumors that are initiated by p53 deletion in the MCA carcinogen. We take these tumors and then make cell lines that could be transplanted into syngeneic mice. The primary tumors are resistant to the combination of radiation therapy and anti-PD-1 therapy. However, the transplant tumors with the same treatment have an immune-mediated tumor cure that requires CD8 T cells. And I didn't show this today, but if we deplete the CD8 T cells, then we no longer get anti-PD-1 radiation cure. The response to tumor transplantation is that if the donor mouse gets its own tumor back, there's immune tolerance to the tumor cells. But if the primary tumor is made into a cell line into a naive syngeneic mouse, there's a robust anti-tumor immune response. And then our exome and RNA sequencing showed that there was high tumor mutation burden in both models, but neoantigen expression downregulation specifically in the primary model. And comparing our mouse models to human sarcomas at the RNA sequencing level, we see that the primary model appears to be most similar to many of the human sarcomas we see, but that the transplanted tumor model may be more similar to the sarcoma immune class E, potentially the anti-PD-1 responders. And single cell profiling I haven't been able to show you today, but again, this is all published, but we see many more activated CD8 T cells in the transplant model, and they're excluded from the primary tumor system. So finally, I just want to take a step back and think about how this may relate to several of the ongoing clinical trials that have recently been published. Here's some data published by Jonathan Schoenfeld and colleagues who's at Dana Farber, looking at anti-PD-L1, anti-CTLA-4, either alone or with radiation. So these are radiation regimens that have been tested in transplanted preclinical models showing efficacy. In this case, this was a clinical trial in patients with metastatic non-small cell lung cancer that had been refractory to previous PD-L1 therapy. And so patients were treated to one site, either with half a gray twice a day for 2 days, so a total of 2 gray to 1 metastatic lesion, or treated to 8 gray times 3 to 1 metastatic lesion. Here are the progression-free survival and overall survival curves, and you can see they're all on top of each other. And so these patients are kind of responding like our primary sarcoma model here, not the transplant tumor models that many have published on, where they see this marked synergy. And so I think that our primary tumor models, I think, are really a gold standard for studying radiation and immunotherapy and other immunotherapy approaches. And we need to develop ways to change the tumor microenvironment so that those kinds of tumors can be responsive to our immunotherapies. So with that, I just want to thank our team and collaborators for all of this work. The first part of this presentation on myxoid liposarcoma was done by Mark Chen. Yvonne Mowry generated the PP3-MCA model, and Amy Wisdom did all of the work characterizing the response of the transplant and primary tumors to radiation immunotherapy. We had a great collaboration for the myxoid liposarcoma work with Ian Davis at UNC Chapel Hill, and our colleagues at Merck and Stanford have been great in the Stand Up to Cancer SARC-32 studies. And with that, I'll be happy to take questions.
Thank you, David, for that amazing talk. Really took us through a couple of really neat stories with a lot of translational importance. I do encourage people to type their questions into the Q&A section down below. We're especially interested in questions from trainees, so please don't be shy. But we do have a few questions that already came in, so I'd love to get your thoughts on these. First of all, what is the minimum RT dose that induces measurable fused CHOP phosphorylation by DDR kinases?
Wow, what a great question. So I don't think we know the answer to that. I think most of the experiments that we did in our in vitro system were with 5 gray or 10 gray. So we didn't kind of do a titration down to see if 1 gray or a half a gray could do that. I do think that it's important to note that low doses of radiation activate the DNA damage response kinases. So we would expect 1 gray or 2 gray could do that, but we haven't actually done a careful analysis to answer that question.
Actually, as a follow-up, do you know if any of those chromatin remodelers, the genes that are associated with fused CHOP, how involved they are with DNA damage responses, and whether you're hindering those to some extent as well when you after you phosphorylate the fused CHOP?
So Chris, I just want to make sure I understand the question. So you're wondering whether the changes in gene expression may sensitize subsequent rounds of radiation. Like there may be impaired DNA damage response based on the changes in the gene expression profile.
Yeah,
that is a
exactly.
that's a good question. So we I don't think we looked at that specifically, but we'll have to go back and look at the data. That's all published and out there, so we can both go back and look at it after the presentation. But I think that's certainly a plausible mechanism that could further contribute to the radiosensitization.
Neat. Another question is, great talk. Given that IR-induced phosphorylation of fused CHOP interferes with tumor growth invasion, would you expect that inhibiting DDR kinases such as ATM or DNA PK may partially counteract the anti-tumor effect of RT for these tumors?
Yeah, Kent, what a great question. So there are now drugs that are really potent and specific inhibitors of either ATM or DNA PK. And these are being tested in combination with radiation therapy right now in various clinical trials around the world. And the idea would be that if you blocked kinases that are needed to orchestrate the DNA damage response, that you should sensitize tumors to radiation therapy. It's interesting. We've done some genetic studies in various mouse models. So we have a mouse model of diffuse intrinsic pontine glioma, where if we mutate P53, then when we delete ATM, they're really radiosensitized. But then if the P53 is wild type in those tumors, and we delete ATM, then we don't see marked radiosensitization. So I think there's certainly a possibility that different tumor types may respond differently to the combination of ATM or DNA PK inhibition and radiation therapy. And this myxoid liposarcoma could be one example, certainly because these tumors are so radiosensitive on their own. We don't really need to give them a radiosensitizer because we're down to 18 gray, and that's giving us 100% local control, and maybe even less would be okay. But I think you're right. That would be really an interesting experiment to block the phosphorylation of fused CHOP, and maybe radiation would work less well.
All right, thank you for that answer. Another question, do other less toxic agents besides radiation induce phosphorylation of fused CHOP fusion protein?
So that's something that we haven't looked at. I think it's likely that other treatments that cause DNA damage will induce phosphorylation. So, chemotherapy like doxorubicin, or other chemotherapy drugs like cyclophosphamide that are commonly used to treat sarcomas that can cause DNA damage to activate these DNA damage response kinases. They'll all probably induce fused CHOP phosphorylation, but there are many other ways to cause stress to a cell. And there might be ways that are less toxic, that don't maybe have the same risk of a secondary cancer down the line. And I think that would be a really interesting kind of direction to take this kind of work, to see what agents may be able to, I think one thing that would be interesting to also look at would be like trabectedin, which is a drug that's active in myxoid liposarcoma, and it can alter transcription. And maybe that's another causing a different type of stress that doesn't cause the same kind of DNA breaks. And maybe that's a mechanism for why that's such a potent drug in myxoid liposarcoma. So I think certainly it would be very interesting to look at that.
Neat. Okay, next question from John Schoenfeld. Fantastic talk. In your sarcoma trials, how are you planning to try to tease out positive immunologic effects mediated by radiation versus potential detrimental effects of a more prolonged radiation course?
Yeah, thanks for that great question, Jonathan. So, when we designed the trial now, it's over 5-6 years ago, we kind of talked about using a hypofractionated radiation regimen of five doses of radiation instead of 25 to kind of address this specific point. So, there's as your point kind of brings up, there's multiple variations, right? There's, when should the pembrolizumab be given relative to the radiation, what dose of radiation is optimal, how many treatments are optimal. And at the time, the radiation oncology sarcoma community was uncomfortable kind of making two changes, changing our radiation dose and scheme and adding in immunotherapy. So we kind of went with the standard of care, historical 25 fractions of 2 gray per day treatment, but we certainly could be killing T cells with each daily dose of radiation. That's possible. We because we didn't know, we gave pembrolizumab before, during, and after radiation and after surgery. Hopefully it'll help at some point. But if we do see that it helps, then the next step will be, is it the neoadjuvant part that's responding so well, or is it because of the adjuvant? Like there'll be a lot more follow-up questions to look at. But I think it's if our data is negative, I don't think it rules out the possibility that radiation could work with pembrolizumab, but maybe we need to give fewer doses of radiation, and that might be a different approach in the future for another trial.
Thank you for that. A couple more questions that we probably have a couple more minutes to go through these. From Freddie Escoria, do you have any thoughts regarding development of small molecule inhibitors to the oncogenic fusion proteins? It appears that on the whole, fusion proteins are where these precision oncology drugs have had their best response. How would you use RT using this approach?
Wow, another great question, Freddie. I think that you can imagine a scenario where you have a drug that maybe binds specifically to the phospho-fus CHOP and kind of freezes it in place. Could be one way to kind of think about that. I think this is a really critical area, and there are a number of outstanding labs that are really trying to use degraders, for example, as a way to target these oncoproteins. I think that's another, like, independent of radiation, a really important direction for the field to go.
Okay, one additional question from James Lorenz. Is there a difference in type 1 interferon response between the transplanted and primary tumors in response to RT plus immune checkpoint inhibitors?
Yes, there is.
Quick answer. And one final question. You've really done a great job showing the difference between these autochthonous tumors versus the transplanted ones. And you're highlighting the issue that a lot of the xenografted tumors that we're utilizing for checkpoint inhibitor efficacy studies in mice may be overestimating the response that we'll get in patients. Do you think that's something that's fairly unique to sarcomas, or is that just going to be something that's seen across the board? And are we kind of overestimating things in everything?
Well, I think what I would say is that all models have pitfalls. I think that the transplant models, you're really vaccinating the tumor, the syngeneic mouse to your tumor cell line. And that doesn't happen in a human patient under most circumstances. So I do think that it applies more broadly than just the sarcoma data that I've shared with you today. But I think that ideally we would do experiments in transplant models and then repeat them in autochthonous models to see, what's the response of our novel therapies in a setting where the cancer co-evolves with the immune system. And probably the most important thing is that we really need to work on our autochthonous models and figure out ways to convert the cold microenvironment into a T-cell rich environment that can be more responsive. I think it's it's a really high bar to get a tumor like this to respond to immunotherapy. And I think if we can do that, the chance for translation, I think, is high.
Excellent. Thank you for sharing your thoughts on that. So we are a couple minutes after the hour, so we will have to cut it off there. But thank you so much again, David, for sharing your incredible research here with us today in this Connect to Science series. We're looking forward to additional speakers in the upcoming weeks. So as I mentioned at the beginning of the seminar, please do check out the Connect to Science website for upcoming links to and Zoom talks. Thank you all so much for joining us today. Thank you again, David, and I hope you all have a great day.
Thanks so much. Bye-bye.